Stop, you're scaring me. You're scaring me.
Orange cats have been sitting in laboratories, living rooms, and shelters for as long as domestic cats have existed. The color is one of the most common patterns in the domestic cat population. Researchers had known since the 1950s that the gene responsible was located on the X chromosome, which explained certain patterns in who could be orange and who couldn't. But for over 70 years after that initial discovery, nobody knew which specific gene it was. The identity of the gene that makes a cat orange remained one of the more embarrassing open questions in animal genetics. Embarrassing because orange cats were everywhere, the inheritance pattern was well understood, and the tools for finding the gene had been available for decades. The answer, when it finally came in 2023, turned out to be hiding in an entirely different field of science. The search took as long as it did, partly because of how genetics research tends to work. Scientists identify a gene by looking for it in a specific context, usually by studying animals or people with and without the trait, and narrowing down which region of the genome differs between them. For orange cats, the X chromosome location was known, which helped narrow the field. But the relevant section of the X chromosome contains many genes, and identifying which one was responsible required either a very large dataset of cats with known color and known genetics, or a specific insight about what kind of gene to look for. Neither came quickly. Pigmentation research in cats focused on other color genes first, because the inheritance patterns of those genes were more complex, and the practical applications seemed more immediate. The orange gene sat in the background, identified by location, but not by identity, for decade after decade. The location of the gene on the X chromosome has a consequence that most cat owners have noticed without knowing why. Males have one X chromosome and one Y chromosome. Females have two X chromosomes. The orange gene, sitting on the X chromosome, operates on a simple principle. One functioning copy is enough to produce orange coloration. A male cat with only one X chromosome either has the orange gene or doesn't. If he has it, he's orange. A female cat with two X chromosomes needs the orange gene on both of them to be fully orange. If she has the orange gene on one X chromosome and a non-orange gene on the other, something more complicated happens. The two X chromosomes compete, and the result is a cat with patches of orange and patches of another color, which is what produces the tortoiseshell and calico patterns that appear almost exclusively in females. Roughly 80% of orange cats are male as a direct consequence of this chromosomal arithmetic. That 80% figure held up consistently across populations of domestic cats from different parts of the world and across different breeds. Wherever orange cats appeared, the sex ratio skewed heavily male. The few female orange cats that existed required both their X chromosomes to carry the orange gene, which means both of their parents had to carry it, which is statistically less common. The consistency of the sex ratio across populations was one of the clearest confirmations that a single X-linked gene was responsible for orange coloration, rather than a combination of genes. The pattern was unambiguous. Finding the gene itself was the problem. The tortoiseshell and calico patterns that result from the two X chromosome dynamic are worth examining because they illustrate something fundamental about how the orange gene operates. In female cats with one orange and one non-orange X chromosome, the body doesn't simply average the two signals and produce a blended intermediate color. Instead, each individual skin cell independently switches off one of the two X chromosomes early in development, a process that happens randomly across the body. The cells that switched off the X carrying the orange gene produce dark pigment. The cells that switched off the X carrying the non-orange gene produce orange pigment. The result is a mosaic. The cat's coat is a map of which chromosome happened to go silent in which patch of skin during embryonic development. No two tortoiseshell or calico cats have identical patterns because the random silencing process produces a different map every time. The orange gene's chromosomal location is what makes this mosaic possible, and it's why the pattern appears almost exclusively in females. In 2023, a research team published the identification of the specific gene. They had worked through a large cohort of cats with known coat colors and mapped the differences in their genomes at high resolution, narrowing the candidate region on the X chromosome until only one gene remained as the plausible cause of orange coloration. The gene they found was called ARHGAP36. The name is not important in itself, but what the gene does is. When the researchers identified ARHGAP36 as the orange gene, the finding landed with a particular kind of surprise. It was a gene that had appeared in research papers for years in contexts that had nothing to do with cats or with color. The gene identified in 2023 was not unknown to science. It appeared in papers on cell signaling. It had been flagged as relevant to certain hormonal pathways. Researchers who worked on ARHGAP36 in those contexts knew it as a regulatory protein, something that switches other cellular processes on or off depending on what signals the cell is receiving. When the cat genetics team identified it as the gene responsible for orange coloration, it was a genuine surprise to scientists in both fields. The cell signaling researchers hadn't been thinking about cat color. The connection between the two fields appeared through a comparison that nobody had designed specifically to find it. The way the orange mutation works involves a cellular signaling pathway that has nothing to do with pigmentation at first glance. Cells in cat skin that produce fur color have a default choice between making dark pigment or light pigment. The signals that tip that choice one way or the other run through a pathway that regulates many other processes in the cell as well. The specific mutation in the orange version of ARHGAP36 alters how this pathway operates in pigment-producing cells, permanently shifting the default toward light orange-yellow pigment and away from dark pigment. The mutation doesn't invent a new biological process. It hijacks an existing one. The same signaling machinery that does something else entirely in brain cells and hormonal cells gets redirected in skin cells to control the color of the fur that grows out of them. The researchers confirmed this by running a series of tests, introducing the orange mutation into cells that normally produce dark pigment and watching the pigment output shift toward orange, then removing the mutation and watching it shift back. The mechanism was clean and specific. One mutation in one gene, expressed in skin cells, rerouted a signaling pathway and changed the color of the fur. The finding was precise enough to explain not just why some cats are orange, but why the orange pattern is so consistent across different cat breeds with otherwise very different genetic backgrounds. The same mutation in the same gene produces orange coloration, whether the cat is a Persian or a domestic shorthair or anything else, because the underlying pathway it hijacks is the same in all of them. The 2023 study also addressed why the mutation produces orange specifically rather than some other light color. Pigment-producing cells have a range of possible outputs depending on which signals they receive. They can make black, various shades of brown, and a spectrum running from deep orange to pale yellow. The ARHGAP36 mutation doesn't simply turn off dark pigment production and leave cells without instruction. It redirects the signaling pathway toward a specific type of light pigment, pheomelanin, which is the molecular compound responsible for orange and yellow colors in mammalian hair. The same pigment is responsible for red and auburn hair in humans and for yellow and cream coloration in many other animals. The mutation's specificity in directing cells toward pheomelanin rather than simply away from dark pigment explains why orange cats are orange rather than white or cream, and it gave researchers a precise molecular explanation for a color pattern that had been described empirically for decades without a mechanistic basis. When the team ran the orange gene against the genetic basis of every other domestic cat color pattern, the differences were stark. Most other color patterns in domestic cats involve genes that are much more straightforwardly connected to pigmentation biology. The genes that produce tabby stripes, the genes that dilute colors from black to gray or from orange to cream, the genes that control white spotting. All of these had been identified previously, and all of them sit in genomic regions associated with pigment cell development or pigment chemistry. ARHGAP36 doesn't fit that category. It's a signaling gene that was recruited into color production through a mutation that altered where and how it's expressed. The comparison revealed that orange coloration in cats is mechanistically different from every other color pattern in the domestic cat genome. Among the roughly 37 wild cat species on Earth, orange coloration is essentially absent. Wild felids come in a range of patterns: stripes, spots, rosettes, and solid dark colors. Some species have lighter undersides, and a few show warm tawny or rufous tones in their coats. But the kind of vivid orange seen in domestic tabby cats, covering the entire body with orange pigment as the dominant color, doesn't appear in any wild cat species under natural conditions. The orange mutation exists in wild cat populations at very low frequencies in some species, but it's rare and tends not to persist across generations because orange is a poor camouflage color for an ambush predator in most environments. A solid orange cat stands out against forest undergrowth, grassland, and rocky terrain. Natural selection removes the mutation from wild populations before it can spread. The near absence of orange in wild cats and its prevalence in domestic cats points to something that happened during domestication. The mutation presumably existed in ancestral cat populations at some low frequency, the same way rare mutations always exist in large populations. In wild cats, it disappeared quickly because orange cats were less effective hunters or more visible to larger predators. In cats living near human settlements, the calculus changed. Humans who found orange cats distinctive or appealing may have preferentially fed or sheltered them. Or simply, the reduced predation pressure and reliable food supply near humans removed the survival penalty for being an unusual color. Whatever the mechanism, once the orange mutation stopped being eliminated by selection, it could spread. The domestic cat population accumulated orange individuals in a way that no wild cat population has, and the gene frequency climbed from rare to common over the 10,000 years of domestication. The timing of when the orange mutation became common in domestic cat populations is something the genome comparison helped clarify. Ancient cat DNA from archaeological sites shows that orange coloration was relatively rare in early domestic cat populations from the Middle East and North Africa. It became more common in medieval European cats, and historical records suggest that orange cats appeared frequently in trade routes and port cities before spreading globally. The pattern of spread tracks human movement, which is consistent with how domestic cat genetics in general dispersed across the world. But the orange mutation spread faster and further than many other color variants. Researchers studying ancient cat genomes found that orange frequency increased in populations wherever cats had been living with humans for longer periods, which supports the idea that human proximity was the key variable allowing the mutation to accumulate rather than being selected out. Multiple studies have looked at whether coat color correlates with behavior in cats. The results are inconsistent enough that no single study can be taken as definitive, but a pattern does appear across several independent research programs. Orange cats are more frequently described by their owners as friendly, tolerant, and calm compared to cats of other colors. Studies that used veterinary assessments rather than owner reports found similar trends, with orange cats showing lower stress responses in clinical settings and fewer aggressive interactions with unfamiliar humans. The mechanism behind a behavioral correlation is where researchers disagree. Some argue that people treat orange cats differently from birth, which shapes behavior through experience rather than genetics. Others point to the gene itself as a potential explanation. ARHGAP36 is expressed in tissues beyond the skin. The same gene that controls orange coloration through its activity in skin cells is also active in brain tissue and in the cells of the adrenal glands, which regulate stress hormone production. The mutation that produces orange fur changes how the gene operates in skin cells specifically. But whether that same mutation in the genetic background of a domestic cat also subtly alters gene expression in neural or hormonal tissue is a question researchers are actively investigating. If ARHGAP36 influences the signaling pathways in adrenal or brain cells even slightly, it could produce a measurable difference in how orange cats process stress or regulate arousal, which would provide a genetic mechanism for the behavioral patterns that owner surveys and veterinary studies have repeatedly found. The connection is not proven, but the gene's activity in neural tissue makes it biologically plausible in a way that it wouldn't be for a straightforward pigmentation gene. The behavioral research on orange cats runs into a methodological problem that makes strong conclusions difficult. When owners rate their cats' personalities, they're not doing so blind to the cat's color. The orange cat is right there, visibly orange, and decades of cultural associations with orange cats as friendly or easygoing may influence how owners describe their animal's behavior. Studies that tried to control for this by using trained assessors who interacted with cats in standardized conditions found smaller but still present differences between orange cats and cats of other colors in stress response and tolerance of handling. The fact that the difference appears even in controlled assessments suggests it isn't entirely a product of owner bias, but it also doesn't prove a genetic cause. The behavioral question remains open. The identification of ARHGAP36 gave it a plausible genetic mechanism for the first time, which is why researchers are now designing studies specifically to test whether the gene's expression in neural tissue produces measurable physiological differences. The human parallel is instructive here. The gene responsible for red hair in humans, MC1R, is also expressed in brain tissue and has been associated in some studies with differences in pain sensitivity and response to anesthesia. The mechanism is similar in structure to what researchers are now investigating in orange cats. A gene that visibly affects pigmentation also has activity in neural pathways, and that dual activity produces effects beyond the color of the hair or fur. The genes are different, and the specific pathways involved are different, but the principle that a pigmentation gene can carry behavioral or physiological secondary effects through its expression in non-skin tissues is established in humans and is being examined in cats following the 2023 identification of ARHGAP36. Comparing orange cat DNA to every other color pattern produced a picture that went well beyond a simple pigmentation story. The comparison confirmed that orange is genetically distinct from all other domestic cat colors in its mechanism. It showed that the gene responsible was hiding in a field of research nobody thought to connect to cat color until the comparison forced the connection. It raised questions about behavior and neuroscience that the discovery of a standard pigmentation gene would never have prompted. And it provided a genetic basis for the observation that orange cats are unusually common in domestic populations compared to wild ones, connecting the gene frequency to the process of domestication in a way that hadn't been mapped before. A color that seems like a simple surface feature turned out to carry a genetic story that runs considerably deeper than the fur it produces. The cat that started all of this was named Cinnamon. The 2007 sequencing of her genome, which produced the first complete domestic cat reference sequence, was what made the 2023 discovery possible. Cinnamon herself was an Abyssinian, a breed that comes in warm ticked coats rather than solid orange. But the genome she provided contained the X chromosome region where ARHGAP36 sits. 16 years after her genome was sequenced, researchers used the reference she provided to locate the gene that had been missing for 7 decades. The orange cats that are currently sleeping on couches and knocking things off shelves in households around the world carry a mutation in a gene that researchers associate with cancer biology, brain development, and hormonal signaling. Nobody predicted that. The comparison found it anyway.